in vivo. If your system isn't mimicking what's going on in vivo, it may be very, very interesting, but it doesn't have as much relevance, as much value as it would. So that's why it's worth focusing on in vivo. I'll give you an example of that in just a minute. The next thing is it can also help you guide you towards reaching your goals. And both of these things then will increase the value of your research. Hopefully you'll get cited more often because your research is of value and is what you're aiming for. So why benchmark? When cell culture started initially, the focus was on getting cells to grow very fast. So let's look at this. In tissues, cells are proliferating very slowly. So, for example, in the hepatocyte, the hepatocytes double in numbers every 200 to 300 days. And that's to replace the cells that have died. So that's quite slow. If we take a hepatocellular carcinoma, so cancer cells, they'll be doubling more or less the same speed. Let's call it 150 to 200 days. A little bit fast, not much. If you take the cell line, HepG2, C3a, in culture, it can double every day. So that is incredibly fast. And that's been the focus of people in the very beginning to get their cells to proliferate. But if you think about it, the fact that they double, let's say 200 times faster, even in the cancer, means that cells in culture are not a good model for the cancer, or for that matter, for the tissue itself. So this is one of the most important points about 3D cell culture. If you take the same cell line, HepG2, C3a, you grow them in the same incubator with the same media, with everything else the same, but you grow them as 3D instead of 2D, then their growth rate slows down. And after 42 days in culture, they have a doubling in numbers of 69 days. So they've got much, much closer to the tissue or the cancer. And therefore, just from that point, you can expect that they'll resemble the tissue or the cancer much, much better. You could, of course, argue that if the cells are growing very, very slowly, then they're dying. The point is that if you look at the ATP content is actually one of the best indicators of cell viability. Then you can see that C3a spheroids grown in 3D conditions, their ATP content goes up as the spheroid gets a bit older. So it shows that the ATP level is increasing. That means that at least apoptosis is not occurring. So when you're doing 3D, you'll wind up sooner or later having to make some assumptions because you're needing to compare to in vivo. So you need some definitions. So some of the assumptions you could use would be, for example, that an average young male weighs 77 kilo and then his liver is 2% of the body weight, and hepatocytes are 70% of the liver by weight, and so on. You'll find you'll need these assumptions simply to be able to relate your in vivo work or the in vivo observations with your research work. You'll also need a fixed point to relate to. So the cells in culture, wet weight would be extremely difficult to measure the weight of the cells in a media. DNA content would be an alternative, a good alternative. So it's quite easy to measure. But the thing is that it varies during the cell cycle. So if you know your cells are all cycling at the same speed, then it's okay. If you compare, let's say, the liver with cells in 2D, then they're in two totally different states in the cell culture, so they have different amounts of DNA. So one easy solution is actually just to measure protein content. There are many ways of doing it, and you should remember that you're measuring just the total soluble protein. There are some proteins that simply will not dissolve in any buffer, and so they are excluded from the measurement. But it's easy enough to define total soluble protein. And in our research, we actually found that we could correlate the total protein content to the area of the cell cluster. So we simply measured the area of our spheroid, and we could then correlate how much protein was present in the spheroid to that area. That's very valuable because you can just take a picture of your spheroid and then you know how much protein you have. So you haven't destroyed it, you haven't done anything to it. It can be still swimming around happily in media. But you know exactly how much protein you have, and you can then put it over into whatever acid you would like. So that's a very convenient and fast way of doing it. The next point I want to make is that giving the concentration is actually, it's like defining how many bricks you have to move, but not defining how many men you'll have to be moving it. So it's only giving half of the story. You can't use concentration for your active molecule, you need to use dose. So when animals are treated with drugs, they're given, for example, a certain number of milligrams of the drug per kilogram of body weight. This is important. I want to give you an example where I actually I did a literature survey and found all of the publications where they compared the LC50 for paracetamol, so that's acetaminophen over in the States. And I found that I found 17 publications. The maximum ratios, the ratio of the highest LC50 to the lowest LC50 was 218. So imagine you go to one doctor and he says, okay, you need to take one tablet to get rid of your headache. And you're not happy with that answer. So you go to a second doctor and he says, oh, no, you need to take 218 tablets to get rid of the headache. That's a terrible ratio. But it's actually not quite true. By making a few assumptions, as I described before, and converting the LC50 values that were written in the publications to the LD50, so the lethal dose 50, the maximum ratio was reduced from 218 to only 20. And I think 20 is a good number, considering that the systems were very varied and the assays were different. Many of different labs and different times and all the rest of it. So giving LD50 immediately makes your results more comparable with everybody else's. So stick to LD50. It's a little bit of extra work to make the assumptions, but in the long run, it'll be well worth it. So that was the few benchmarking tools we need to get into place, and let's see what you can do with them. In our Hep2 C3a spheroids, we measured the production of cholesterol. And as you can see, we're giving cholesterol in micromoles produced per day per gram of total protein. And we measured it as the spheroids were getting older, and you can see that it increased, but it reached some sort of plateau after about 18 days. So the spheroids are 18 days old. We did the same sort of study for urea production. We know that the Hep2 cells are missing the enzymes in the urea cycle, but they use the alternative pathway. And there, they also can produce urea at a level which is similar to the liver. And that plateau also is reached about 18 days. Now, We know that many cell lines need, let's say, about 18 days to reach this plateau. It may not be the same for stem cells or primary cells. Stem cells may need much less time, or they may never be able to recover the functional activity of the tissue you want to look at. Primary cells probably are at in vivo levels at the beginning, but the cells are dying. And so they may be actually losing their ability. So it's important to measure the parameter you want to look at with time. So the question is, why do cells need this time? In culture, cells are communicating like crazy. They're doing it in many different ways, and they're very, very busy at doing it. In 2D cell culture, unfortunately, people use trypsin every, let's say, 4 to 5 days. And the trypsin destroys the cell communication. It also destroys cell orientation and gene regulation, the infrastructure of the cell, the extracellular matrix, and a lot of other things. So really, trypsin is not, or other enzymes, is not a good thing to use. Another reason for changing to 3D. In 3D, you never treat the spheroids or organoids with trypsin, and so you allow them to communicate. And so they're highly connected and highly busy at talking together. Another example of spheroids mimicking in vivo behavior is this one here. Here we've treated our spheroids, we've grown them in 2 concentrations of glucose, 5.5 millimolar or 11. So 5.5 would be a normal glucose level, whereas 11 would be a diabetic level. And we see that spheroids grown at 5.5, you can treat them with insulin, and then they increase their glucose uptake and then produce glycogen. At 11 millimolar, that physiological response has disappeared. So the spheroids become glucose resistant, just like a patient would in vivo. So here we can mimic a disease. Another example we've done is this one here. Here I've taken again a literature survey, and on the horizontal axis, I've plotted the in vivo lethal plasma concentration for 6 common drugs. And then on the vertical axis, I've again done a survey of all the literature available for these 6 drugs and calculated the average LD50 drug concentration. And so if the in vitro and in vivo were perfectly correlated, it would be a straight line and all the spots would lie on the line. And you can see that that's not the case, that the coefficient of correlation, the R squared, is only 0.55. So there is a correlation, but it's not very good. Those studies were done in 2D. You can do the same studies using human hepatocytes, so freshly derived primary human hepatocytes. And for the same 6 drugs, there the R squared then increases to 0.75. So what the pharmaceutical industries are doing using human hepatocytes is obviously much, much better than growing cells in 2D. If you take those cells grown in 2D but grow them in 3D, as in our studies, then the correlation coefficient increases again. So an ordinary cancer cell line grown as spheroids in 3D is a much better predictive marker for toxicity in vivo than primary human hepatocytes. So I hope this shows the power of a very simple system, a very reproducible system, a fairly inexpensive system, in using it for clinical research, drug development. Here's another example. Here we've taken our 3D spheroids and we've given them various doses of paracetamol, acetaminophen. Here I'm showing the dose where one single dose is enough to kill the spheroids outright. So the little black arrows at the bottom of the screen indicate when we are treating the cells. These spheroids are already 21 days old at the start of the experiment. So 0 is 21-day-old spheroid, and we treat them there, and then at 48 hours, 96, and so on. And you can see that one dose at this top concentration kills them outright. If we halve the dose, then we can see that the cells survive longer, and you actually need to treat them 4 times before you kill them. So before we had an acute lethal dose, here now we have a chronic lethal dose. If we halve the dose again, then we don't kill the cells. The cells, the spheroids are responding, and they seem to be learning and responding stronger over time, but the doses are not lethal. And if we then go down a bit further in concentration, now down to the normal doses that would be given to somebody with a headache, you can see the cells are still responding, but of course it's not lethal. So here's an example of an in vitro system that can be used to test repetitive drug treatments. And this offers an alternative to treating animals. And this is now also based on human cells. So it's a valuable alternative to the treatment, to the possibilities that are existing at the moment. Now I want to present some work by others, just to show that it's not just us, but many other people are beginning to get very, very exciting results using 3D spheroids or organoids. Here's some results using 3D clusters of INS1 cells. These are from the islets of Langerhans. And they've shown in their publication that cells will respond to glucose concentrations by synthesizing insulin and amylin. So this is in diabetes research. It's some nice work there. Chris Nagals presented this last week, gave a very nice presentation showing that LS180 colon cancer cells respond in a similar manner to that seen in patients and can also develop drug resistance. So again, mimicking in vivo physiology. Here's an example of work done by Young and co-workers, where they've been looking at primary cells and then their differentiation into dental pulp. We're limited time, so we'll stop there, but go on maybe to something that's equally important, because then you can say, well, what is the secret of getting in vivo-like results? And that's maybe a question many people ask. Well, the answer is pretty simple. There isn't a secret. You don't need to use special media or growth factors or cytokines or other factors. You don't need matrigel. You don't need to find a special batch of matrigel, a good one. You don't need it at all. You normally don't need collagens or laminins or anything else special. All you need to do is to grow your cells in whatever you are currently using. And that's true for primary cells, stem cells, induced pluripotent stem cells, or cell lines, that grow them as clusters, spheroids organized in suspension for a few days in the normal media you use. If your differentiation process needs growth factors, then of course you add them, but I'm trying to stress that you should repeat what you're doing normally, but just do it in 3D. So if you want to do it in 3D, there's a couple of ways of producing your spheroids or organoids. The first one is simply to put the cells into what we call the Kleiner reactor, I'll show you that in a minute, as a single cell suspension, and then wait for them to form organoids or spheroids. The organoids or spheroids produced will be of different sizes, and that could be a limitation if you want to be very precise in your work, but this is a very straightforward way of doing it. If you want organoids or spheroids of very similar size, then you should use low adherent microtiter plates. In our studies, we can produce spheroids this way, and 21 days later, when they've had a chance to mature and settle down, the variation in protein content is only 22%. So they are very, very uniform. And that means you can pick out a couple of spheroids, and then a few days later pick out another couple of spheroids, and they'll be very, very comparable. A third way of producing your spheroids or organoids is simply to stick them together with something like alginate or matrigel or collagens or laminins. Here we're not using these things to change the performance of the cells. We're simply using it as glue to hold the cells together. So there are some cell lines or cells that don't want to make a spheroid or organoid, and you can then force them to do it by using a little bit of glue. Once you've got your spheroids, you can put them into the Kleiner reactor. Here's the Kleiner reactors that we're producing. There's a 10 milliliter culture chamber in here, and then this lid goes on top. So you can see through it. So you can just take this Kleiner reactor, put it under your microscope, and look at it. Look at the spheroids you've got without disturbing them. Around it, there are some beads, these blue beads, and they simply provide humidification for the culture. And that's a trick we use to reduce the risk of infection. Your culture times are longer in 3D, and so that's why it's good to try and reduce the risk of infection.
Steve?
Yes.
There's something with your voice. Can you get closer the microphone?
I can try.
Good. Thank you.
My face will probably be very big on the screen now, but I'm very close to it. Is that any better, Torsten?
Yes, I think so.
Okay. When you put your spheroids in the Kleiner reactor, then you put the Kleiner reactor in this instrument we've developed, the Kleiner star. And basically, you just put them in here, and then this rotates. So you can put 6 different cultures in. It's a CO2 incubator, so it keeps the temperature and CO2 very constant. In the door, you've got 6 cameras, so you can look at your cultures as they are going. So this is what it looks like in vivo. All those little pale yellow spots are liver spheroids. Notice, for example, how uniform in size they are. They are moving very slowly, so that means at least that nutrients and waste products can be exchanged quite easily with the spheroids. So here's the workflow. It's the workflow for immortal cell lines, but it's not very, very different if you have primary cells. You get your cell suspension, you make your spheroids by one of the 3 methods we talk about, transfer them to the Kleiner reactor, put the Kleiner reactor in a Kleiner star, and then for cell lines, as I described earlier, you may need to cultivate them for 18 days so they can recover from trips and damage. They can establish good communication between each other, And then you can start your experiment out here. And as also mentioned earlier, stem cells or primary cells may not need this 18-day recovery period. You have to find out what it is for your cell line, your cells. Because of this gentle rotation, you can keep your clusters alive for weeks or months or even years. So that's very valuable. The clusters can get very big, a millimeter in diameter or even larger, depending on the type of cell you're working with. It's because of the gentle movement that the media moves across the surface that enhances the diffusion. So we call it active diffusion. But actually all it's doing is to make the diffusion gradient short. That's a very important gain and allows your surrogates to become so large. Kleinerstats have been used for growing hundreds of cell lines already during the last 20 years or so since they've been first introduced. So these cell clusters have been benchmarked and shown to mimic the in vivo phenotype in many, many situations. And it's therefore possible to produce very valuable results for your research, and hopefully this will then enhance your careers. So the simple take-home message is you want to let your cells do what they're programmed to do. Just give them the best conditions to do it in. And by saying let your cells do what they're programmed to do, you don't want to influence them with external scaffolds or external growth factors or unknown compounds. You just give them enough time to settle down, make contact with each other, and then they will work out themselves what they want to do. And that will probably bring you faster, closer to the in vivo situation. I hope the sound quality was good enough, and I would love to answer any questions that anybody might have. Thank you very much for listening.
Thank you, Steve. I hope that most of you could hear most of Steve's talks, at least throughout the presentation here. So if you have any questions, please use the Q&A box. We already got a few. So the first question, Steve, is how can we estimate the relation between our model and in vivo situations?
There you have to pick what are you actually wanting to measure. I mean, the in vivo situation is a very vague term. So if you wanted to look at something like cholesterol synthesis, you need to set up a cholesterol assay, estimate what cholesterol synthesis is in vivo, and then use your assay to see if your cell lines or organoids are doing what they should be doing.
Good. Thank you very much, Steve. I hope that answers the question. I have another question here coming up is if I work with stem cells, how long could I keep them in the bioreactor? And how will I know that they are ready or if they have become too old?
Nice question. Again, by benchmarking against an in vivo functionality, you'll know when they're ready. If you are kind and remember to change the media, then you can keep them alive for, as I said, over a year. So you have plenty of working time that way around. Of course, if it's a primary cell line, they may be dying. So you may be limited by the cells, but not by the culture system. So again, use your benchmark, follow the functionality you want to study, and see when it is comparable to that that's happening in vivo. And there you have your system.
Okay, thanks a lot, Steve. I have another one. How can this machine control the organoid's size? And what if the center of the cells are dead? What if you have a necrotic core?
There's a lot of good questions coming. Certainly, it's not a good idea to have a necrotic core. So your second part of the question leads back to your first. Yes, one way of controlling the size of your organoids is simply to vary the number of organoids you have in the Kleiner star, in the Kleiner reactor. If you have very few organoids, they will tend to grow very large. If you have a lot of organoids, they will be smaller. And the explanation is quite simple, that the cells are very active in the Kleiner reactor. And so there will be a competition for food. And that is actually something that helps stimulate recovery to in vivo conditions. So by regulating the number of organoids you have, you can regulate their size.
Okay, that makes sense. Also for me, Steve. Thanks. So I have another one here. Can I adopt, is the workflow similar to all constructs grown in the system?
Essentially, yes. It's a very flexible workflow. Obviously, if you're starting with biopsies, you can put biopsies straight into the system, and they should be functional from day one. So that would be a very short workflow. Whereas if you've had to dissect out cells, produce a single cell suspension, then it'll be much longer. But it's a very simple, straightforward workflow, but I think many people should easily be able to follow.
Okay, thank you. Then I have a question here says, does dissociation method influence recovery time? Trypsin or alternatives are available? Trypsin and alternatives are available. So that's what the question is. Does dissociation method influence recovery time? Because there's alternatives to trypsin.
There are alternatives to trypsin, yes. I think the recovery time is proportional to the amount of damage you do. So if you've, let's say, scraped off cells from a adherent plate and then call those your spheroids, that should probably be the minimum amount of damage. So they would be expected to be recovered much faster. So it's proportional to the damage you do. So whatever enzyme you use, if it's causing a lot of damage, you'll have a longer recovery time, and vice versa.
Okay, thanks, Steve.
You're very polite, Torsten. You'd think we didn't really know each other.
But I'm a polite man most of the time, at least. So
okay.
Then I have another question here. So what measures can we put in place for organoids that are not uniform in shape? For instance, not spheres.
Right. We've designed the Kleiner reactor so that it's actually quite easy to open the lid. So in the worst case, if you've got large organoids, you could open the lid and pick out the ones you like or the ones you don't like. And in that way work towards a more uniform population. If the organoids are small, maybe you can even work through one of the ports. But the easy way is to open it up, and then you have a Petri dish and work that way. The beads, actually, the humidification beads, then act as a temperature buffer and help to keep your cells warm and cozy, even though you're in that noisy, sterile bench, sorting them out.
Actually, I have another question regarding the bleeds. So it's what are the blue balls surrounding, or what are the materials of the blue beads surrounding the Kleiner reactor or the incubation chamber?
Essentially, they're polyacrylamide beads. I think they're used widely also in arranging plants and things. So they're just spherical polyacrylamide balls, which are then hydrated with sterile water. So no magic there either. And they are, because it's polyacrylamide, then it should be totally polymerized and there's no leakage of anything toxic into the cells.
Good, thanks. Yeah. Then another question here is, I'm sorry if this has been brought up in the PowerPoint before, but is the proliferation rate for the spheroid as long as in vivo cells?
As you saw from one of the early slides, after 42 days, the proliferation rate was a doubling time of 69 days. So no, we didn't get down to a doubling rate of 200 days. We went for a sort of useful compromise so that we knew that the functionality of the spheroids was comparable to that was happening in vivo. And so we decided to start our experiments as a standard after 21 days only. It's a balance. You don't really want to wait, let's say, 3 months before starting your experiments. It takes some work keeping the spheroids happy and alive.
Good. Then actually I have a question from Poland, from Simone. He asks, do you see the tendency for spheroids to combine to aggregate of the number in Kleiner star is quite high?
That can happen. If you have too many spheroids in there, then they can stick. It'll depend on your cell line. But yes, if they start to stick together, then try and dilute them. That'll, of course, mean that they begin to get a bit larger again. But the tendency to aggregate is worse at the beginning, I think, than when they get settled down. So avoid too many, and after that, hopefully your problem disappears.
Okay, thanks, Steve. Then I have another question. Is it possible to do co-cultures in the system?
Yes, excellent question. I didn't touch on it at all. It is very possible and very straightforward. You could make your co-cultures in many ways, either by mixing the cells from the beginning and let them form the spheroid, or make a spheroid and then coat it with another cell line or many other variants. It's an excellent thing to do. Obviously, it becomes a bit more complicated. You have to make sure your growth rates of the cells so that one cell line doesn't overgrow another, but you can recover additional physiological properties by doing co-culture. So super question. And yes, you certainly can.
Good, thanks. Then I have follow-up questions. Any particular recommendations when working with primary cells to develop organoids?
There, I think no particular recommendation because it'll depend on the cell type. But I think your benchmarking will become ultra important because primary cells, unfortunately, have a tendency to die. So you need to find the best time with which to work with them. They're making maybe the alginate spheroids could give you a kickstart because you produce the alginate spheroids very quickly, and maybe then you can work faster. You could save a few days that way.
Good. I think, that was most of our questions.
They've been excellent questions. I enjoyed it.
Yeah, that's good. Yeah, I'm really pleased with all the good questions from our participants here today. So I think I will close the webinar now, and thank you very much, Steve. I think you also have your email here and your contact information. So if anyone has some additional questions to Steve, please feel free to reach out to Steve. You can also always reach out to Cellvivo at info@cellvivo.com. And besides that, I will wish the ones from Europe a good day, the ones from Asia and Australia a good evening, and a very, very good early morning to the US people. So thank you very much, Steve, and
thank you all for listening, and thank you for the questions.
Okay, thanks. Bye.
Bye-bye.